Assalamu alaikum, guys. Good evening. What we were discussing last time was the process of transcription, and we have gone in detail about the transcription process and also have gone through in detail about the role of messenger RNA and in eukaryotic cells, the post transcriptional modification which is needed. Now, the ultimate goal of that transcription is to get the final product, and the final product is protein. So, for this transcription that resulted in the formation of the messenger RNA, whether it's a prokaryotic messenger RNA where there is no requirement of post transcriptional modification, or it is a eukaryotic process where there was post-transcriptional modification, including the capping on the 5' and a poly A tail on the 3' end, and the process of splicing, where the introns are being left inside the nucleus while the exons coding the exact gene will be there in the final product. So this mRNA, which is being synthesized as a result of the transcription, will now uh, be used for the next step, and that is called translation, which is in short being called as protein synthesis. Now, for the translation, there are two more RNA molecule which will come into action. One of them is called the ribosomal RNA, which is a structural component of ribosome, and we do have the difference of the ribosome uh, in terms of the eukaryotes and the prokaryotes, but I'm not going in that detail at this stage. And there is another RNA which is the tRNA, or the transfer or translator RNA. So these two RNA will now be into action to read what actually is being coded on the mRNA to get the desired product, the protein. So the translation of messenger RNA into protein will be, as I said, accomplished by the ribosome. That means ribosome is the site where the protein synthesis takes place. And ribosome itself is a RNA protein hybrid. And the ribosomes are composed of two subunits, a larger unit and a smaller unit. Now that's common in both pro and eukaryotes, but the sedimentation coefficient of these units are different in case of the pro and eukaryotes. So ribosomes will then bind to the translation initiation sequence on the mRNA. So the mRNA molecule has got a specific sequences by which it's going to be attached with the ribosome, and then it will move down the RNA in the direction of 5' to 3'. As a result of this, the reading of the genetic code on the mRNA will be read from 5' to 3' direction. And as a result of that, the codons, the specific amino acids will be coming in and forming a polypeptide chain. Now, the first amino acid on the polypeptide chain will always have a free amino group, because the next incoming amino acid is going to attach to the carboxylic side of the amino group. So if you start a polypeptide chain, the first amino acid of the polypeptide chain will always have a free amino group, while the last of the amino acid of the polypeptide chain would have a free carboxylate group. So this is a must condition. So this particular site with a free amino group is being called as N-terminal, while the other side where the last amino acid would have, the free carboxylate group will be called as the C-terminal. So the growing polypeptide chain, as a result of the translation, would have one N-terminal and the other one is called the C-terminal. Now, each group of the three nucleotides present on the mRNA, which we know are called as codon, and every codon refers to one amino acid. We all know that. So a transfer RNA will come into action, because transfer RNA is basically an adapter. This has got anticodon on this, and as a result of that anticodon, the transfer RNA can read the codon present on the mRNA and then will bring about the corresponding amino acid. So we discussed last time about this picture of the transfer RNA. This is a clover leaf-like structure. Uh, just make sure to see that this transfer RNA is also a single-stranded molecule, although at some places there are hydrogen bondings, which has resulted in the formation of four loops in the tRNA structure, or giving it a particular clover leaf-like structure, but it is considered as a single-stranded molecule. So we have this amino acid loop, we have this anticodon loop, and then we have two more loops. Here we are not that interested in going in the detail, but these two loops where we have anticodon that read the frame on the mRNA, and the amino acid loop or site where the amino acid is attached. So each tRNA has its own corresponding amino acid. There is a concept called the Wobble hypothesis. The Wobble hypothesis say that this three anticodon here, which actually read the frame, one of that anticodon is variable. That means the third base of the anticodon is called the Wobble base, and it can form base pair with several different nucleotides, thereby giving it the option to read more than one frames. So that's the one thing that you need to just keep it in mind. Now, going in the detail of the translation, just like the transcription, we divide that in different stages. Like if you remember in the transcription, the first stage was initiation, where the enzyme will bind and results in the formation of a space where the two DNA strands get separated. Then the next step is the elongation, where the mRNA synthesis takes place. And the last step is the termination, where the mRNA synthesis is complete. Very similarly, we have these three steps in translation: initiation, elongation, and termination. So in the initiation of the translation, in particularly in prokaryotes, the ribosome will bind to the specific translation initiation sites. There can be several different initiation sites present on a messenger RNA molecule. A prokaryotic mRNA can code for several different proteins. So depending upon which specific initiation site is going to attach with the ribosome, it can give rise to different, kind of proteins. So the translation begins at the first codon, or the initiation codon, and that is being called as AUG codon. And in some cases, this could be GUG codon. Now, the modified amino acid N-formyl methionine is always the first amino acid of the new polypeptide. Now, this is a very important point. Like whenever the mRNA is going to attach with the ribosome, it has specific initiation sites. And we're talking about at this stage with reference to the prokaryotic mRNA, and they always have the first codon either AUG or GUG. This codon will always, code for an unusual amino acid. This is not a normal 20 amino acid. It's an unusual amino acid, which is called N-formyl methionine. So whenever the polypeptide chain is growing, or whenever the translation process is starting, the first of the amino acid coming in always will be N-formyl methionine. In eukaryotes, the ribosomes bind to the 5' cap, which you already know that in the messenger RNA of the eukaryotes, there was a post-transcriptional modification, and there was a 5' cap called 7-methyl guanosine. So in eukaryotes, the ribosome always bind to the 5' cap. So now you got the understanding that why there was a 5' capping in the eukaryotes, because that helps in the binding to the ribosome. Then move down the mRNA until they reach the first AUG codon for methionine. So that's the difference of a pro and a eukaryote. Now the translation starts from the point specifically, which is called the initiation site. The eukaryotic mRNA code for only a single gene. Now, in case of the prokaryotes, as I said, there are multiple initiation sites. So based on that, one prokaryotic mRNA can code for several proteins. While in case of the eukaryotes, the mRNA code for only one single gene, right? Note that translation does not start at the first base of the mRNA. There is an untranslated region at the beginning of the mRNA, which is 5' untranslated region. So keeping in mind that this mRNA is still having some regions on it which are not the part of the final protein product actually. So this is what we call the 5' UTR, or the untranslated region. Now let's go in the detail of the initiation. As you can see the picture, it's step by step, letting you know. You can see this ribosome, this larger unit and a smaller unit. Initially, the larger and smaller unit are separate. Once there is a binding of the messenger RNA to the smaller unit, it will bring about the reunion of the large and the small subunits. So the initiation process involves first joining the messenger RNA with the initiation sequence, or what we call the methionine tRNA, which will bring the first amino acid, which is always methionine in case of the eukaryotes, and the small ribosomal unit is the one where it attaches. Now, several initiation factors are also included in this. There could be many proteins which are involved, just like in the initiation of the DNA replication, initiation of the transcription, and they do have their roles in the binding of this mRNA to the smaller subunit. And as I said, once it is bound, it will bring up with a large subunit, and then it's joined to make a complex, just like this one. And this is what the first, the tRNA, which is bringing the methionine, which is always the first amino acid in case of the eukaryotic translation process. Now, once this complex is being formed, that's all about the initiation, because that's how the initiation starts. After this, the process will be carried on further, and the next step of this is called elongation. Now, what happens in elongation actually, the ribosome has got two sites for the tRNA. One site is called P and the other side is called A. Now, this is very important and very tricky, so you have to be a little bit focused on that. The tRNA, can bind on the ribosome on two different sites, because once the mRNA is attached with the ribosome, now the tRNA will come into action and they start reading the frame of the messenger RNA, and accordingly will bring about the respective amino acids. So the point is, when the tRNA is coming, it's going to attach, or it's going to combine with the ribosome on two different sites, or you can say there are two sites for that, what we call P and A site. Now, what the function of these sites are and what is their role? Now, the initial tRNA with attached amino acid is attached on the P site, while a new tRNA corresponding to the next codon, according to the gene sequence on the mRNA, will bind to the A site. Now, remember there are two sites, P site and A site. What I'm saying that the first tRNA, which is coming and bringing amino acid, will attach to the P site, while the next tRNA coming with the next amino acid will bind to the A site. The ribosome will then catalyze a reaction. And what this reaction is? It transfer of the amino acid from the P side onto the amino acid at the A side. Remember, the first tRNA was there on the P side, and the next tRNA coming in is on the A side. What is happening actually? It's working like that the ribosome will catalyze the transfer. Now, this amino acid, which is on the P side, will be taken away, and then it's going to attach with that amino acid, which is on the A side. And that's how a dipeptide unit is formed. Now, the question arises here that why it is like this? What's the significance of this? Now, the significance of this can be understood that, as we know that the first amino acid has attached on the P side, the second amino acid has attached on the A side, and then the ribosome has catalyzed the formation or the transfer of amino acid from the P side to the A side, and thereby leaving another space available for the next molecule to come in. we'll discuss that later on when we'll go in the further detail. So just to make sure at this point that the first amino acid coming from the first tRNA will be coming on the P side. The next one will be coming to the A site, and then the ribosome is going to transfer the amino acid from the P site to the A site, thereby making a dipeptide unit. Now, the next step is the ribosome will then move down because now we have to read the whole mRNA. First codon has been read. So it's just like, if you look at this picture here again, you can better understand it's going like this one, it moves across this one, and it's going to read this genetic code one by one. So here you can see that the ribosome then moves down one codon, and the now empty tRNA at the P site is displaced of the ribosome, and the tRNA that has the growing peptide chain on it is moved from A site to the P site. So that means this dipeptide which has been formed will now move from A site to the P site, and thereby you can see that the A site is now empty to get another tRNA coming in and bringing in another amino acid. So the process is then repeated. So what happens actually, the tRNA at the P site will hold the peptide chain, and always the new tRNA coming in with the new amino acid will bind to the A site. Then there will be a transfer reaction. This amino acid is going to be transferred from A site to the already existing chain on the P site, and then the A site is empty again to receive another tRNA. And that's how the process goes on. The reading frame is read and new tRNA coming in all the time, attaching on the A side, and then ribosome will catalyze the reaction to transfer that amino acid from the A side to the growing chain on the P side. So the chain is growing on that side, and new amino acid from the tRNA are coming on the A side. And that's how the process goes on till it reaches the point where the entire reading frame is being read and all the amino acids have been brought in, and as a result of this the chain is complete. Now this picture is going to give you an idea. This is the initiation, and now you can see that the reading frame is going on and we are coming in with the more and more amino acids. So this is the site from where the new amino acid is coming, and this is the site from the new amino acid is leaving. So this is what we call the P site, where we have the growing polypeptide chain, and this is the A site where the new amino acid comes in, give its amino acid. This amino acid move from there to there, and then this will leave. So every time the amino acid transfer takes place, the tRNA that has brought in that amino acid will leave the A site, and the new amino acid with the new tRNA will be coming in to the site. Ultimately, as I said, the whole reading frame has been read, and there comes the process of termination. Now we know that there are three codons which are the stop codons or the nonsense codon. They code for no amino acid, and all protein coding regions end at a stop codon. So when the ribosome reaches a stop codon, then there is no tRNA for that. Instead, protein called release factor is come into action, and the name itself indicating the release factor. What is the function of this protein? It's going to release the mRNA, or in other words, it's going to dissociate the mRNA from the ribosome. And that means dissociation of the mRNA from the ribosome means the process is completed. You can see both smaller and larger units get separated, and this is what we have, the protein chain which is being formed here, and this is what we call the dissociation factor, or in other case this is the release factor, which is being brought in as a result of the stop codon, which gives the signal that the process is complete. Note that the mRNA which continues the path of the stop codon, the remaining portion is not translated because that's not part of the gene, and that's how it is called the untranslated region. So we have untranslated region which is being shown here and here. 5' end and 3' end both have untranslated region which is not the part of the gene actually. So just like the transcription, there are certain changes, there are certain amendments after the process of translation is complete, and we call them post-translational modification. So now we have a polypeptide chain, and this polypeptide chain will undergo certain changes. And what are these changes? That is what we call the post-translational modification. Now, the new polypeptide usually fold themselves spontaneously because we know that straight chain is unstable, and that attains an active conformation. However, in some proteins, they are being helped and guided in their folding process by certain other proteins, and we call them chaperone proteins. So some proteins will fold automatically, and some proteins might require the support of other proteins for their folding, and these support proteins are called chaperone. Now, many proteins have sometimes added sugar group or phosphate group or fatty acid or other kind of covalently attached molecules. And this is what one of the post-translational modification is. You have studied about the proteins, you have studied about the carbohydrates, and you have studied about the lipids, that they are complex. So lipids can combine with protein, protein combine with carbohydrates, and so on and so forth. So these kind of changes, like glycoproteins, for example, they have protein and the carbohydrate moiety. So this is what we call the post-translational sort of modification, and that is being mostly done in the endoplasmic reticulum, and this is mainly done in eukaryotes. Many proteins are targeted for a specific site, so they need to be transported to different organelles within the cell. And this targeting is accomplished through different sort of signal sequences which are being on the polypeptide chain. In the case of the proteins that go to the endoplasmic reticulum, the signal sequence is a group of amino acids at the end terminal of the polypeptide, which are then removed from the final product, because the purpose of that specific a group of amino acid was only the transportation. They're not the part of the final protein product. So once it reaches the site, they are no more needed, and they will be cut down. So after that, we will end up today's lecture with a little bit of discussion of genetic code. I'm not going to, discuss much because we already have had the discussion of the genetic code in our lecture of Introduction to Molecular Biology. So we all know that genetic code is a set of three nucleotides which code for one, gene, one protein, sorry, one amino acid, and then there are 64 genetic codes, out of which we have 61 functionally active genetic code that code for amino acids. So there are 20 amino acids. So we have more than one genetic code for one amino acid. Then there are three stop codons, which we call the nonsense codon or the terminating codon, that we already have studied. And this is the figure of 64 genetic codes. And you can see these are the three: UAA, UAG, and UGA. These are being called as the stop codon. This is another picture of the genetic code. Uh, Something interesting about the genetic code is that the genetic code is almost universal. Like, for example, if let's say AUG code for methionine, it will code for methionine no matter it's a prokaryotes or a eukaryotes, no matter it's a unicellular or a multicellular small organism to a complex one. So this is one very universal characteristic of genetic code. However, some variants exist, specifically in mitochondria, where we have some genes which have these variation of amino acids with a genetic code. For instance, CUA codes for leucine is a universal code, but in yeast mitochondria, it codes for threonine. Similarly, AGA codes for arginine as a universal code, but in human and Drosophila mitochondria, it is a stop codon. So only the case of the mitochondria where we have the exception, rather than the rest of the things would be in terms of a universal code for an amino acid. There are also few known variants in the code used in the nuclei in some of the protists, but we are not going in the detail of that. So with this, we have finished our today's lecture. And with this, we ended up the process of transcription and translation. Inshallah, in the next few classes, we'll be discussing about gene regulation. So I'm going to stop lecture here. And if you have any questions, just raise your hand and ask the question. I'm willing to answer. Thank you. Okay, if there is no question, so I'm going to finish the lecture at this stage, and inshallah, I'll share the recording once it is uploaded. And see you guys on Monday for the practical exam as per the scheduled time. Class CR Plus, please share the message again on the group as a reminder for Monday's practical. Thank you very much, and Khuda Hafiz.